This video will cover the last stage of cell respiration, which is oxidative phosphorylation. Before we cover this, it's worthwhile to remember a few of the basics behind redox reactions. Remember that in organic chemistry, redox reactions follow electrons, but more specifically, you want to follow the hydrogen atoms, right? So a chemical that's being oxidized generally loses hydrogen atoms, and a chemical that's being reduced generally gains those hydrogen atoms. So, for example, when oxygen is reduced by picking up electrons, it's reduced to water. So that's a good thing to keep in mind. Another good thing to remember when we're talking about oxidative phosphorylation is the structure of the mitochondrion. You've heard, for example, where glycolysis and where the Krebs cycle occur, and it's good to remember where oxidative phosphorylation occurs as well. Specifically, we're going to be looking at the inner membrane of the mitochondrion and all its infoldings, which are called cristae. Remember that the space inside the inner membrane is called the matrix, and the space between the inner and the outer membrane of the mitochondrion is called the intermembrane space. So those regions are all going to be important for us as we talk about oxidative phosphorylation. So we have already been through glycolysis, which split glucose in half, and we've been through the Krebs cycle, which continued the breakdown of glucose. At this point, most of the energy from glucose is in energy-rich electrons. Some of it has been used to make some ATP by substrate-level phosphorylation already, but that's a relatively small amount of ATP, right? Most of the energy in glucose is going to be used for our payoff phase in oxidative phosphorylation right now. So this is the exciting phase of cell respiration. It's where we get most of our ATP from that glucose. So how exactly do we do that? Well, oxidative phosphorylation includes two steps. One is the electron transport chain, which explains how we get energy out of those electrons. And the second is chemiosmosis, which explains how we use the energy from those electrons to build ATP. Let's start with the electron transport chain. This diagram is symbolic. It shows four protein complexes, numbered here with Roman numerals, which show various electron carriers. The electrons will be dropped off by the electron carriers NADH and FADH2. Now remember, like anything else, once NADH and FADH2 drop off their electrons, they get oxidized, and NADH goes back to being NAD+, FADH2 goes back to being FAD. Fine, so they've dropped off their electrons, they're good. They can actually go back to earlier stages of cell respiration and now pick up more electrons to continue driving this process. But they've dropped off their electrons, and now those electrons that are high in free energy will jump from carrier to carrier. Now, each of these carriers is a little bit more electronegative, which means each one is a little bit more welcoming to those electrons. The electrons start the process really high in energy. You can think of them as being like really antsy. But every time they get to a new electron carrier, they get a little bit more comfortable. They lose a little bit more of that energy. Eventually, they go all the way down to the most electronegative electron carrier of all, and that is oxygen. Oxygen, highly electronegative. And so those electrons, when they bind to oxygen, they're so relaxed, they're so low in energy, they're so happy to be there. Oxygen, picking up those electrons, becomes reduced and forms water. All right, the electrons are done. They've given up their energy, but what happens to that energy? This diagram here actually shows you the same thing. We see the electron carriers, but now we get a little bit more context for what's going on. This membrane that those electron carriers are embedded in is actually the inner mitochondrial membrane, which makes this space on the bottom the matrix and this space on top the intermembrane space. You'll notice that the matrix is colored a lighter orange and the intermembrane space is colored a darker orange. That's important. This is the case because it's representing a proton gradient. In the lighter area, protons are less concentrated. In the darker area, in the intramembrane space, the protons are more concentrated. Remember that, it's important. All right, so we see here the yellow arrows indicating the flow of electrons, right? They're jumping from carrier to carrier, and we know that they're giving up energy with each jump. Where does that energy go? Well, this diagram shows us that something else is happening at the same time. You see these red arrows. These protein complexes don't just allow electrons to give up their energy. They actually harness that energy to drive protons, to pump them against their electrochemical gradient and push them into the intramembrane space. This is active transport, right? It requires energy. Usually, cells get the energy for active transport from ATP, but here they're getting the energy from the electrons, which, remember, with each jump are losing a little bit of energy. And here in this diagram, we get to see where that energy goes. It's used to pump protons from the matrix into the intramembrane space. These protons are not happy. These protons are now high in energy. There's a lot of potential energy there in this gradient because the protons are just dying to go back into the matrix, right, where they're less concentrated. The thing is, there is only one molecule that will allow those protons to diffuse back into the gradient. Remember, ions can't diffuse easily across a membrane. And so they find this molecule right here, ATP synthase. It's a combination of an enzyme and an ion channel. As an ion channel, it allows protons to diffuse back into the matrix. And those protons are so happy to do that. Once they diffuse into the matrix, they're more relaxed again, they're lower in energy. But they are going through with such kinetic energy that we call the energy that they have as they travel through ATP synthase the proton motive force. It's really energy, it's really not a force, it's really energy. Right? But there's so much energy in those protons as they rush through the ATP synthase to go back into the matrix. There's so much energy there that ATP synthase can immediately harness the energy of those moving protons to drive the endergonic process of attaching an inorganic phosphate to ADP and generate ATP. Now you'll remember, that takes a lot of energy because that third phosphate group does really not want to get attached to the ADP. It really, really doesn't. Okay. But the energy of those moving protons is so great that it can be used to drive that process. And this is where we see the real payoff of cell respiration, right? This is where we generate so many ATPs. It's in this process of chemiosmosis. Osmosis because the protons are diffusing. Chemi because it's something other than water that's diffusing, right? Usually osmosis means the diffusion of water. Chemi is a tip-off that we're talking about diffusion, but it's not the diffusion of water, it's the diffusion of something else, in this case, protons. All right. So, to recap, chemiosmosis summarizes two processes. It's the electron transport chain, where electrons are stripped of their energy, and that energy is used to push protons against their electrochemical gradient. Oxidative phosphorylation also includes chemiosmosis, where those protons rush through ATP synthase by diffusion, and their proton motive force is used as an energy source for driving the synthesis of ATP. This oxidative phosphorylation process is where we get most of the ATP that's generated in cell respiration. That's why it's called the energy payoff phase.